This is me Arzu here, and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 4 video of the development of the teeth, and in this video we will be particularly discussing with the advanced belly stage, and in the previous video we have discussed with the early belly stage. Okay, so we have covered the overview of the development of the teeth in the first video, and body stage and cap stage in the second video, and in third video we have covered the early belly stage, and in this video we are covering the advanced belly stage, and in upcoming video we will cover the physiological stages of the development of the tooth. Okay, so I hope you guys have watched those all the parts of the development of the teeth. Okay, and after that in this video we will be discussing in detail about this advanced belly stage. Okay, so early belly stage was the critical stage of the tooth development. Okay, and in this advanced belly stage and the early belly stage, the main difference is the mineralization. Okay, so sometimes they can ask you in the viva what is the main difference that is between the early belly stage and the advanced belly stage is the mineralization. Okay, so mineralization means there is a formation of the first layer of the dentine, okay, and the first layer of the enamel. Okay, so the mineralization, after the mineralization there is formation of the both the enamel or the dentine. Okay, and there is also the formation of the root. Okay, so these two events are very important in this advanced belly stage. So without delaying much, let's get started. So in advanced belly stage, as I have written here, the mineralization and the root formation. Okay, so these two are the most important events of this advanced belly stage. After that, enamel organ. Okay, it shows the 4 different layers, that is inner enamel epithelial layer, okay, that is forming the ameloblast, then after stratum intermedium, and then after stellate reticulum and outer enamel epithelium. Okay, so these all 4 layers are also present in the early belly stage. Okay, so both the stages are coming under the belly stage. But in the cap stage, if you have remembered, this stratum intermedium was absent there, and similarly in the bud stage there was also absent of this stratum intermedium, as this is absent in the cap stage. Okay, so this stellate reticulum was present in the cap stage, but in bud stage even this stellate reticulum is also absent there, and only the cell that is present in the bud stage is the peripheral cell and the central cell. So the peripheral cells were cubital in shape and the central cells were polygonal. Okay, similarly in this stellate reticulum there were star shaped cell. Okay, and there were outer enamel epithelium, inner enamel epithelium. Outer enamel epithelium was low columnar types of cell. Okay, and similarly inner enamel epithelium was the columnar type of cells, and the outer enamel epithelium was cubital there. Similarly, after that, this stratum intermedium formed in early belly stage, and the stratum intermedium would have squamous cell layers. Okay, 2 to 3 layers of squamous cell is present in this stratum intermedium layer. So I think you have remembered all those points. Okay, after that, in the next point, as I have written here, the cervical portion of the enamel organ give rise to the Hertwig's epithelial root sheath, that is called horse. Okay, so that is called horse, and it is responsible for the shape, length, size, and the number of the roots. Okay, so this is one of the most important point here. Okay, as I have told here, as I have written here, that the development of the root takes place in this advanced belly stage. So this Hertwig epithelial root sheath is the thing that is giving rise to the formation of the root. So cervical portion of the enamel organ give rise to the Hertwig epithelial root sheath and is responsible for the shape, length, size, and the number of the roots. Okay, so I will be discussing with this horse here. Okay, in next paper I have prepared a proper flow chart. Okay, so that you can remember easily, and similarly you can write in the your university level examination. Okay, so now our another point is the reversal of the polarity, and this is one of the most favorite questions. Okay, so in the previous video also, as I have discussed with the reversal of nutrition there. Okay, in early belly stage. Okay, so that was also very important, and in the reversal of nutrition, as I have discussed, okay, so due to the formation of the dentine layer, first dentine layer, the nutrition is cut off from the dental papillae to the inner enamel epithelium, and after that the inner enamel epithelium get closer to the outer enamel epithelium by the collapsing of the stellate reticulum, and it derives its nutrition from the dental follicle cells. Okay, so the connective tissue, that is dental follicle cells. So remember that. Okay, and in this reversal of the polarity here, as I have written here, the nuclei shifts to the proximally and towards the stratum intermedium, and the Golgi centrioles move towards the distal end, that is towards the dental papillae. So I will explain with this point. Okay, so if this is a ameloblast, okay, and previously the ameloblast, okay, and this is a basement membrane of the ameloblast, and here it is dental papillae, okay, and here is a stratum intermedium. Okay, so what happens is that, okay, so what happens is that, so first of all the nucleus is towards the dental papillae side, okay, and the Golgi complex and the centrioles are in the proximal side. Okay, so means proximal side means towards the stratum intermedium. This is very important to understand. We will discuss with this in the amelogenesis. Okay, when we will discuss about the development of the enamel, particularly there we will be discussing with this reversal of polarity, but also as we are discussing with the development of the tooth, this is important here too. So towards the stratum intermedium, it means it is a proximal end. Similarly, towards the dental papillae means it is a distal end. So what happens is there, reversal of polarity means this nucleus which is near the dental papillae moves towards the stratum intermedium, means nuclei shift to the proximal end towards the stratum intermedium, and the Golgi complex and centriole moves towards the dental papillae, and that is means moving towards the distal end. Okay, so just remember those 2 points. Okay, so they are very important. And the significance of this reversal of polarity is that, okay, moving of this Golgi and the centrioles this side, okay, so they would release different kind of proteins. Okay, the enamel proteins they can release. Through those proteins there would be the differentiation of the cells in the dental papillae to the odontoblast. Okay, so that is the significance of this reversal of polarity, but we will be discussing properly in the amelogenesis chapter. Okay, now after that, as I have written here about the reciprocal induction, so these 2 points are important from this advanced Bell stage. Okay, so reciprocal induction means the inner enamel epithelium that is forming the ameloblast. Okay, so we know that inner enamel epithelium, okay, it derives into the ameloblast, it differentiates into the ameloblast. After that there is a release of the enamel proteins and the growth factor by those ameloblast, and those enamel proteins and the growth factor would cause the epithelial and the mesenchymal interaction. Okay, so as I have discussed in my previous video, there I have told, okay, anything in this development of the tooth is happening due to of the epithelial and mesenchymal interaction. Okay, so there is interaction between the connective tissue and the epithelial layer, okay, that is forming different structures. Okay, so this is important to understand here. So there is epithelial and mesenchymal interaction happening. After that it causes the differentiation of the cells of the dental papillae in the odontoblast into the odontoblast, and after the formation of odontoblast it forms the dentin, and there deposits the first layer of the dentin, and it starts from the cuspal areas towards the cervical areas. Okay, cuspal areas means towards the from the cusp, okay, from the cusp tip towards the cervical areas, and I will be explaining this. And after that reciprocally, okay, ameloblast deposit the first layer of the enamel that starts from the cuspal area to the cervical area. Okay, and I have written one note here, that is differentiation and the mineralization all starts from the cusp and move towards the cervical area. So I will explain all this proper flow chart. Okay, so just you listen to me. Okay, so what they are telling to us that inner enamel epithelium. Okay, so I will draw a picture for you. Okay, so I will draw a picture for you here. Okay, so just think. Okay, so this is a inner enamel epithelium. Okay, so this is a inner enamel epithelium, and okay, so here is a dental papillae. Okay, so this is a dental papillae, and here it is a cell free zone. Okay, this is a acellular zone. Okay, so this is a acellular zone. Okay, and this is a inner enamel epithelium here. Okay, and nucleus is present towards the basement membrane, and after the reversal of polarity it moves towards the, so in another picture. Okay, so okay, so nucleus is moving here. Okay, nucleus is moving here, and in this area there is happening this, these are not cells. Okay, these are enamel proteins. Okay, I will just show with another pen, then you will be clear. Okay, so don't get confused, these are the enamel proteins and the growth factors. Okay, so they are the enamel proteins here. Okay, so they are the enamel proteins, and after that these are the cells. Okay, so there is a formation of the odontoblast. Okay, So these are the odontoblastic processes here. Okay, so this is forming like this. Okay, so as I have written here, okay, so I will explain by this picture. Okay, so as I have written here, first of all, this inner enamel epithelium, okay, it is differentiating into the ameloblast, okay, and that is reciprocally done by, okay, this odontoblast. Okay, the thing means, okay, so the thing that I want to explain is that, okay, so the formation of the ameloblast depend upon the formation of the odontoblast, and similarly the formation of the odontoblast depends upon the formation of the ameloblast. Okay, so there is a reciprocal induction. Okay, one induce another and another induce the another. Okay, so the thing that is happening here is that, so the inner enamel epithelium, it forms the ameloblast, and that ameloblast would release the different kinds of the enamel protein, okay, and the growth factor. So these are the enamel proteins and the growth factor in this picture. So those enamel proteins and the growth factor would differentiate those cells, okay, of the dental papillae into the odontoblast. So odontoblasts are formed. Okay, and another thing that you should remember here, that I have written here, it starts from the cuspal area to the cervical area. Okay, so I will show you in the picture of this, okay, advanced bell stage. So as you can see here, okay, so as you can see here, okay, so in this picture, so this dark pink is a enamel, and this reddish like structure, not pink, it is red. Okay, so that is a dentine. Okay, so I mean to say is that, okay, so as you can see, the thickness of dentine or the thickness of enamel is higher in the cusp, and it is decreasing towards the cervical area. Okay, it is moving towards the cervical area. It means that, okay, so the formation of this or the deposition of this enamel or the dentine takes place from the cusp. Okay, so there is a formation of this enamel or the dentine first takes place at the cusp, and after that it moves to the cervical area, and it is similar in the process of differentiation too. Okay, as I have written in the note here, the differentiation and the mineralization both starts from the cusp and move towards the cervical area. Okay, either it is a dentine or in the enamel. Okay, so differentiation means, as I have written here, inner enamel epithelium is differentiating into the ameloblast, so it is first happening at the cusp, and after that it is moving towards the cervical area. Similarly, as I have written here, okay, differentiation of the cells of the dental papillae into the odontoblast, so this is also happening at the cusp, and after that it is happening in the cervical area. So as there is first happening the differentiation on the cusp, so there must be the deposition of the dentine or the first formation of the dentine at the cusp. Okay, so it is of sure that, so that there is a first deposition of the dentine at the cuspal areas, and then it moved towards the cervical area following the path of the differentiation of the cells. And similarly in this ameloblast too. Okay, and reciprocally, so this term is important here. Okay, so reciprocally they both activate each other. Okay, so that is called reciprocal induction, and this is very one of the most important concept in this chapter. Okay, so now we will study about the Hertwig epithelial root sheath and its root formation. Okay, so as I have written here, the development of the root begins after the enamel and the dentine formation has reached the future cemento-enamel junction. Okay, so when the, as I have told that enamel and dentine, okay, so enamel and dentine starts to form from the cusp or the incisal edge, and it moves to a cervical end. Okay, so when it reaches the cemento-enamel junction, at that time this Hertwig epithelial root sheath is formed. Okay, and after that, as I have written here, Hertwig epithelial root sheath is the formed by outer and inner enamel epithelium, and it separates the dental papillae and the dental follicle. Both are cuboidal in shape. Okay, so just watch once this picture. Okay, so in this picture, as I have shown here, this is an inner enamel epithelium, and this is a basement membrane that is separated from the dental papillae by a cellular zone. Okay, where there is just a present of the connective tissue ground substance, but no any cell is present there. Okay, after that, okay, so when this inner enamel epithelium, okay, this inner enamel epithelium, there is reversal of polarity there. This nucleus reaches to the proximal side, and Golgi complexes and centriole comes to the distal side. Okay, so they releases the enamel protein that would cause the differentiation of these cells of the dental papillae and forms the odontoblast with odontoblastic processes as here shown. And this is a ameloblast. Okay, so this inner enamel epithelium self also get induced into the get activated, get differentiated into the ameloblast. Okay, so the same thing is here. So you are clear with this. Now in this figure, as you can see, this is a enamel organ of the cap stage. Okay, so I am just drawing this for the schematic purpose, and this is a rough diagram. Okay, so don't get confused with that. So just think this is a cervical loop. Okay, cervical loop means it is a junction between the outer enamel epithelium and inner enamel epithelium, and I have discussed with this cervical loop in the early Bell stage video. Okay, so when this both the layers of outer enamel epithelium and inner enamel epithelium, okay, they just meet at the cervical loop, and from that cervical loop there comes an extension. Okay, and the extension is of two cells. Okay, so there is two cell layer. Okay, outer is the outer enamel epithelium, inner is the inner enamel epithelium, and it goes growing and it proliferating downside. Okay, so the same thing is written here. So this is formed by the outer and inner enamel epithelium that separates the dental papillae in the side of inner enamel epithelium and dental follicle on the side of outer enamel epithelium, and both are cubital in shape, but when they are present in enamel organ, okay, so at that time the inner enamel epithelium is a columnar type of cells present there, and after that outer enamel epithelium have the cubital type of cells. So after this, okay, so I have prepared a flow chart. Okay, so this flow chart is very important. Okay, so after that what happens is that, so inner enamel epithelium, okay, it sends the signal to the dental papillae, and that results in the differentiation of the cells to the odontoblast. Okay, so the cells that are present in the dental papillae, they would differentiate into the odontoblast. After that, in another point, as I have written there, once differentiation ends, okay, so after the completion of the differentiation, so the cells of the Hertwig epithelial root sheath, they undergo degeneration. Okay, and after the degeneration, so the cells that are remnant, okay, so the remnant cells of this Hertwig epithelial root sheath is called cell rest of molasses. Okay, so this is important to remember, cell rest of molasses. Okay, which is a dormant cell, but it can cause pathology. Okay, so throughout the life it is dormant, but it can cause some kind of pathology. After that, in the fully developed tooth, okay, they are found near the periodontal ligament or near the cementum. Okay, so in fully developed tooth, when we will cut the cut section, and when we will watch that cut section in the microscope, we can see this cell list of molasses, which is dormant cell. Okay, in this near the periodontal ligament near the cementum. Okay, okay, so the periodontal ligament is connecting the cementum and the alveolar bone. So at that position, as we can see this cell list of molasses there. After that, I have written here, after degeneration, dental sac come in the total contact with the dentin and fasten the development of the cementoblast and from the cells of the dental sac. Okay, so as there is degeneration, okay, so just think there is a dental papillae here, and this is a dental follicle here. Okay, so this is dental papillae here, and this is dental follicle here. So after degeneration of this boundary, okay, so the barrier between those two layers, okay, those two portions of the tooth gum, then they would come in contact. Okay, and after this inner enamel epithelium had sent the signal to these cells of the dental papillae, they would differentiate into the odontoblast, and after the formation of the odontoblast, there is degeneration of this. Okay, so after that degeneration, that odontoblastic layer of this dental papillae and this dental follicle, okay, they just come in contact, and after that contact, this odontoblastic layer gives the signal to these cells of dental follicle, and that would forms the cementoblast. Okay, so cementoblast is the formed from the cells of that dental follicle or the dental sac. Okay, after that, in the next point, as I have written here, prior to the root formation, root sheath forms the epithelial diaphragm, outer enamel epithelium and inner enamel epithelium, and bends at the future cemento-enamel junction into the horizontal plane and narrowing the wide cervical opening of the tooth gum. Okay, so as I have discussed here, okay, so Hertwig epithelial root sheath is formed by outer and inner enamel epithelium. So you may be confused that what is this epithelial diaphragm. Okay, so I will clear your concept by showing a picture. So as you can see in this picture, so this is a picture of Orban's. Okay, so this is a book of this is Orban's. Okay, so as you can see here in this picture, okay, so this is a enamel organ here. Okay, so this is this layer that is being shown here, this is a enamel organ. After that, okay, this darkish line is separating this enamel organ from the enamel. Okay, and after that enamel, we can see this darkish portion. Okay, So this darkest portion is the dentin, and the junction between enamel and dentin is the dentino-enamel junction here. So what happens is that, as I have told, from the cervical loop there comes a process. Okay, there is an extension that is called Hertwig epithelial root sheath. So this epithelial diaphragm, as they have written here, so just you should have to clear a simple concept is that, okay, so epithelial diaphragm is a portion of Hertwig epithelial root sheath, but the thing that comes firstly, okay, that comes firstly as an extension is the epithelial diaphragm, okay, not a proper two layer cells of this outer and inner enamel epithelium. It is the epithelial diaphragm of the Hertwig epithelial root sheath. It comes earlierly, and after that, okay, the cell that is in this free end, okay, so this is a free end of this epithelial diaphragm. So the cells that is present in this free end of the epithelial diaphragm, they do not participate in the proliferation, but the cell that are proximal to this epithelial diaphragm, okay, so the cells that are proximal to this epithelial diaphragm, they just proliferate and forms like this structure, okay, so they are forming the root sheath, okay, so the proper layer, okay, so the proper layer of the two cell, okay, is formed by the proliferation of the cell that is proximal to the free end of the epithelial diaphragm. Okay, so that is the main concept you should understand. Okay, so firstly the thing that is formed is epithelial diaphragm. Later the proliferation takes place in the proximal cells of the epithelial diaphragm, which leads to the epithelial diaphragm to move in the downward direction, okay, while this the two layer cell is formed in upward direction, okay, simultaneously, okay. So the same thing written here. So the prior to the root formation, root sheath forms the epithelial diaphragm, outer enamel epithelium and inner enamel epithelium bends at the future cemento-enamel junction into the horizontal plane. Okay, so that is a horizontal plane there, and after that narrowing the wide cervical opening of the tooth gum. Okay, so wide cervical opening. So this is the opening of the tooth gum. Okay, and this epithelial diaphragm is just closing that wide opening. Okay. And after that I have written here, free end of the diaphragm doesn't proliferate, it is the epithelium coronal to the diaphragm, it proliferates. So same thing I have written here. And after that, differential growth of the diaphragm in the multi-rooted teeth causes the division of the root trunk into the 2 or 3 root. Okay, so 2 tongue like extension of the horizontal diaphragm develop in the lower molars and 3 in the upper molars, and those extensions grow toward each other and fuse, and single cervical opening of the coronal enamel is then divided into 2 or 3 openings. Okay, so the thing that is telling here is that, okay, so as you know that, okay, some teeth have one root. Okay, in anterior tooth we can see one root, but in a posterior teeth we can see 2 or 3 root. Okay, similarly in the maxillary tooth, maxillary molars, we can see 3 roots present there, and similarly in the mandibular molar there is just present of the 2 root. Okay, so how these roots are formed? Okay, how more than one roots are formed? Okay, so as I have told here, if there is a formation of this epithelial diaphragm, okay, it's okay, then epithelial diaphragm would form just one single opening, no, but how the 2 or 3 roots are formed there? So that is the thing you should understand here, so that you will be cleared by this diagram, as you can see here. Okay, so just think this is a, okay, so this is a simple picture. Okay, so this is of mandibular molar and this is of maxillary molar. Okay, because there are formation of the 3 roots. So what happens earlierly is that, so there is a proper a concentric circle like structure, means firstly this epithelial diaphragm, okay, so they just come closer and a proper circle like opening is present there. So this is a opening. Okay, so this is the opening of this area, and what happens after that is, so the area that is surrounding this, there happens a differential growth. Okay, so differential growth takes place in this portion. Okay, so that is differential growth, means differential growth is taking place in this epithelial diaphragm. Okay, means that some portion they just don't get proliferated, but some portion get proliferated, as like here. So the area that is here, okay, so they are proliferating and just getting closer to each other, but the area that is present here, okay, so they are not proliferating. So this differential growth takes place there, and there is formation of the tongue like extension. Okay, so those tongue like extension grows and grows, and they just fuse at a single line, forming a two structure. Similarly in a third, 3 types of 3 roots. Okay, So in 3 root, what happens, there is also a differential growth. Okay, so the differential growth is taking place here, and the taking place here. Okay, so there develops a tongue like extension in that area, and those tongue like extension just get meet, and there is a formation of the 3 roots. Okay, so that is a simple concept you should understand. Okay, and in this picture, as you can see here, okay, so this is structure. So this structure is this, okay, or this. So this is opening, okay, this is a apical foramen, and this is a bifurcation. Okay, so there is a bifurcation of the both the roots. Okay, so I think you have been cleared in this concept. So just I will recite 1 second. So what happens is that firstly there is the extension formation. Okay, so there is a formation of the extension from the cervical loop, and cervical loop is a junction between the outer enamel epithelium and the inner enamel epithelium. Okay, so that extension firstly which is formed is a epithelial diaphragm, and the cell that is proximal to this epithelial diaphragm. Okay, so the cell that is proximal earlier to the epithelial diaphragm, they just get proliferated and they just form the layer. Okay, and that layer is a Hertwig epithelial root sheath. But don't get confused that that layer is only a epithelial root sheath. Okay, it is not so. The epithelial root sheath means the layer to cell layer and also that epithelial diaphragm. Okay, so I have written here, it is outer enamel epithelium, and that is formed when it is reached at the, so as I have written in this point, development of the root begins after the enamel ending formation as is the future cemento-enamel junction. Okay, so just have like that concept, and in this portion as I have discussed, there is happening a differential growth. Okay, so that differential growth is causing the formation of the tongue like extension, and those tongue like extension thus get fused. Okay, and after fusing there is a formation of the two, so there is just a partition in the single circle. Okay, so those partition would divide the single circle into the different circles. Okay, and that is forming the apical foramen. Okay, so that is the proper concept here, you should understand. Okay, so same thing I have shown here in the schematic diagram. So this, the first thing that is formed is epithelial diaphragm, and after that epithelial diaphragm the extension is happening in the direction that is proximal to the epithelial diaphragm. Okay, and there is a formation of the Hertwig epithelial root sheath, and this is a epithelial diaphragm here. So okay, now in another point as I have written, if the continuity of the Hertwig epithelial root sheath is broken or is not established prior to the dentine formation, defect in the dentinal wall, then there is the development of the accessory root canal. Okay, so what happened is that, okay, so as when I will be discussing with the pulp, okay, so in pulp chapter I will be discussing with this accessory root canals. Okay, so we know that, okay, if this is a anterior tooth, okay, and this is a root here. Okay, so this is a apical foramen present here, this is a apical foramen, but sometimes what happens is that, okay, some hole are present in the areas of this. Okay, so this is a pulp. Okay, so this is a, I have drawn, this is a schematic diagram of pulp. Okay, so this is pulp. So what happens is that in the radicular pulp, okay, so in the root portion of the pulp, sometime the apical foramen as it opens in the apex, okay, from it knobs and vessels goes inside the pulp, but also sometimes the opening also present in the radicular pulp. Okay, so that radicular pulp opening is called accessory root canals, and that is formed by the continuity of horse is broken or not established prior to the dentine formation. Okay, so as you know that dentine is formed, and after the dentine, okay, formation, after the formation of odontoblast, there is a breaking or the continuity, there is a degeneration of the Hertwig epithelial root sheath. But if the degeneration takes place previously the formation of the dentine formation, at that time there is a development of the accessory root canals. Okay, so that is the thing we should remember. After that I have written here, if the cells of the Hertwig epithelial root sheath remain adherent to the dentine surface, they may differentiate in the fully functioning ameloblast and produce the enamel. Okay, so this is important to understand here, and such droplets of enamel is called enamel pods. Okay, so this much for today, and we will meet in the next video, in the next part of this development of the tooth, where we will discuss with the physiological stages of the tooth development. So stay happy, stay healthy, and stay tuned. Till then, tata, bye bye, thank you.